Now we're going to pause for a few minutes in our look at the actual pathway that food takes through the digestive system to look at how specific nutrients are digested in the different compartments. So we're going to go through each of the 4 types of biological macromolecules and look at how and where they are broken down. We're going to start with carbohydrates. For these molecules, we start with polysaccharides and disaccharides and break them down into monosaccharides. This occurs in the oral cavity via salivary amylases that we already talked about. And it also occurs in the small intestine, that's what SI stands for, via pancreatic amylases and disaccaridases. So these amylases are breaking down your bigger polysaccharides into smaller ones, ultimately to disaccharides, and then your disaccaridases are the enzymes that are breaking down those disaccharides into monosaccharides. 2 things I want to note before we get to part B here is that 1, for each of these slides, I'll have some enzymes highlighted. I'll tell you the degree to which you need to know what those enzymes are doing. So like for carbohydrates, you need to know that salivary amylases break down carbohydrates not all the way to monosaccharides, but from big polysaccharides to smaller ones and to disaccharides in the mouth. And then any enzymes that are not highlighted in red, you're not going to be responsible for those specific things. The second thing I'll point out is how this figure works. On each of these slides, it's figure 4112. So we're breaking up the digestive system by compartments. So we have mouth, stomach, and then both of these boxes represent the small intestine. It's just are the enzymes coming from the pancreas or the small intestine itself. You can just treat these as one box because we're never going to ask you to tell us specifically whether a small intestine enzyme comes from the pancreas or the small intestine. We're just going to lump all those together and say they're active in the small intestine. So the compartments here are mouth, stomach, which doesn't do anything for polysaccharides, and then the small intestine. The next macromolecule in line is proteins. So we're taking our polypeptides and we're breaking them down to amino acids. Protein digestion starts in the stomach, and we've already talked about the activity of pepsin, which generates smaller polypeptides. And then once you get to the small intestine, you have a number of enzymes, pancreatic trypsin and chymotrypsin, dipeptidases, which break down dipeptides, 2 amino acids, into single amino acids. There's also carboxypeptidases and aminopeptidases, which break down polypeptides from either end, 1 amino acid at a time. The enzyme I need you to know here is pepsin. You should recognize like carboxypeptidase as something that breaks down polypeptides, but you're not going to be required to tell us anything else about that. Our third macromolecule is nucleic acids. So what we're doing here is taking our DNA and RNA and breaking that down into the component parts of the nucleotides. So we're not just going from a polymer to the monomers, we're actually breaking those monomers down even further. So we're generating nitrogenous bases, sugars, and phosphate groups. Nucleic acid digestion occurs entirely within the small intestine. The only enzyme name I want you to really know here is nucleases. Nucleases are the main enzymes that are breaking down nucleic acids. Then there's a whole bunch of other enzymes that are breaking down those nucleotides into their component parts, nucleotidases, nucleosidases, and phosphatases. As far as these 3 go, I want you to be able to recognize those as enzymes that are involved in nucleic acid digestion, but other than that, you don't need to be able to tell me specifically what they do, just be able to recognize which macromolecule they digest. And then finally, we have lipids, which are usually ingested as large molecules called triacylglycerols, or triglycerides is the more common name for those molecules. Now we have a problem here, is that these molecules are hydrophobic, but enzymes are water-soluble, and we're going to be digesting them in an aqueous solution. So these hydrophobic molecules are going to do what? Exactly, they're going to clump up, which makes it hard for the enzymes to get in there and break them down. The solution to this is bile salts from the liver. Our liver makes these materials called bile salts, which are then stored in the gallbladder and released into the small intestine during digestion. Those materials are going to emulsify the lipids, basically break up those big masses of lipids into smaller chunks, which have a greater surface area. This allows the enzymes to more efficiently break them into smaller pieces. You can actually model this process of emulsification yourself if you're doing the dishes. So you take a little bit of oil in a pan, for example, and you put some water in there, and you'll see that the oil will form like droplets. It'll form little bubbles in the water because oil and water don't mix, as the saying goes, because the oil is hydrophobic. If you put a little bit of dish soap in there, that's going to emulsify that oil so that it's going to break up. And what'll happen, and you'll see it kind of like shoot across the surface of the water very fast, what'll happen is it'll turn those bubbles, it'll break them down into little bits of oil, and it'll actually make that water kind of cloudy because the oil is distributed through it. That's the process of emulsification that the bile salts accomplish in the small intestine. In other semesters, I would joke that, haha, you're not doing your dishes, you're college students, but now with everybody home, maybe you are doing dishes. So if you get a chance, model emulsification in your kitchen and teach your family about how lipids are digested in the small intestine. The enzymes that are accomplishing this are called lipases, so they're going to break those triglycerides into the components, the glycerol, the fatty acids, and in some cases monoglyceride, so a glycerol with a single fatty acid tail instead of 3. So here's your summary for macromolecule digestion. We have our carbohydrates, proteins, nucleic acids, and fats. We have the carbohydrates being digested in the mouth and the small intestine, proteins in the stomach and the small intestine, and the nucleic acids and fats just being digested in the small intestine.